We have Alex Marson, who's at the University of California, San Francisco, and he's going to be talking to us about reprogramming human T-cell circuitry.
Well, thank you for having me here today. Good morning. The overall goal of research in my lab is to understand genetic circuits that control the human immune system, and in particular, genetic circuits that control human T cells. We're interested in how these circuits control healthy function and how they fail in disease states. And then ultimately, what we want is to develop new therapies that target the underlying causes of circuit failure in a range of human diseases. And advances in genome engineering now open up the possibility that we can really ask fundamental questions about these genetic circuits directly in primary human immune cells. But as we've developed tools to do genome engineering and start pulling apart these circuits, we've also realized that there's an opportunity here to directly engineer T cells themselves as a new class of cellular therapies. And that's what I want to talk to you about today, is really opportunities to develop cell therapies combined with genome engineering. And in order to fully realize the potential here, this is what we envision we really need. We need the ability to take human immune cells out of patients and then have complete flexibility over how we engineer specific sites in the genome of these cells. We've heard a lot about opportunities to excise pathogenic sequences from the genome, potentially remove checkpoints. But what we want is flexibility beyond that to not only cut sequences out of the genome, but actually replace particular mutations that are causing disease, and even beyond that, insert large new sequences, not into random sites in the genome, but into specified endogenous sequences. And these later applications of replacing mutations and especially inserting large sequences into specified sites have really been a technical challenge, and that's what I think we've made some advances that I want to emphasize today. So just to go back a few years, when I first started the lab, our goal was to bring CRISPR directly into primary human immune cells, and early on there were very quickly applications in model organisms and cell lines. But when we went to start to put CRISPR into T cells, we and others hit a series of roadblocks, and the fundamental one was really a problem of delivery. And when we tried to use nucleic acid-based delivery, especially of the Cas9, to primary human T cells, we had essentially no success. And the turning point for us was a wonderful collaboration with Jennifer Doudna's lab, where we fundamentally changed how we were delivering the Cas9 system. Instead of putting plasmids or lentivirus encoding the Cas9, we directly took purified Cas9 protein, loaded it in vitro with guide RNAs to make a preformed ribonucleoprotein complex, or RNP. And what we found right away was that these RNPs could be directly electroporated into primary human T cells, and immediately we saw high efficiency editing. And beyond that, we realized that this was very attractive from a therapeutic standpoint. We were delivering a protein complex that would be transient, it would be degraded over time, but it would leave a permanent modification at the site where it cut the genome. And that would permanently modify the genetic program in the targeted cells. The other advantage that we came to realize was that because we had very tight temporal control over when this double-stranded break was going to be introduced, we could co-deliver, we could co-electroporate a DNA repair template along with the Cas9 RNP. And when we did so, we found that we could not only introduce indels to knock out a gene, but we could actually use homology-directed repair to rewrite specific nucleotides at a specific site in the genome. So to step back, the tools that we developed, we had developed these Cas9 RNPs, which were very powerful for gene knockout and genetic testing directly in primary human T cells. We found that we could co-deliver the Cas9 RNPs with single-stranded oligos, the 200 base pair oligos, or 200 base oligos that could be co-delivered with an RNP. And this would allow us to rewrite individual nucleotides, but it didn't give us much real estate to actually manipulate sequences. And there have been recent advances in now trying to develop viral-based templates. So several groups, including Andy Scherenberg, Matt Porteous, most recently Michelle Sadelain, have used non-integrating or largely non-integrating forms of AAV as homology-directed repair template, where the viral genome itself is used as the template and combined with Cas9 ribonuclear proteins or other nucleases. And this has led to some pretty impressive results of knocking in large sequences, but it comes with the liability that it requires manufacturing a virus for each repair that you want. And what we've realized was that in the lab, if we wanted complete flexibility to test a lot of sequences, we wanted to avoid this reliance on virus. And the need to avoid virus is even more acute when you think about translating this to therapeutic applications, where each virus would have to be made in a GMP-compatible fashion. So the challenge that we set for ourselves was, could we move away from virus completely? Could we combine the Cas9 RNPs directly with a DNA repair template? And what we and many others had observed was that DNA at concentrations sufficient to think about it, using it as a repair template, is highly toxic to T cells. And that had really stymied this field. But Theo Roth is a pretty spectacular MD PhD student in my lab. He happens to be here in the audience, but I think I would say that anyway. He's a pretty spectacular MD PhD student in the lab who was undeterred by the conventional wisdom that DNA was excessively toxic to T cells. And he set out on really a heroic attempt to try to systematically test different variables that may allow us to introduce the combination of Cas9 RNP and DNA repair templates into T cells to enable high efficiency gene editing while maintaining cell viability. So this is what listed the set of conditions that he began to test. He tested a range of different cell culture and stimulation conditions, a range of different concentrations of the ribonuclear protein and concentrations of the DNA template, and very importantly, ratios between those two. He also tested different electroporation conditions, and we took advantage of a 96 well plate electroporation format that we have established in the lab. And that allowed Theo to test really hundreds of different combinations of conditions, all the while optimizing for knock-in efficiency and cell viability. This was the construct that he used for these experiments. This was a construct designed to knock in a GFP fusion tag into a ubiquitously expressed housekeeping gene, RAB11. So he was trying to insert 750 base pairs of the GFP using 300 base pair homology arms on either side of this DNA template. And if successful, the cells should be green as a result of this fusion protein. So this was the result that really floored me. Theo was actually able to get GFP expression in the majority of cells using this, and at the same time maintaining high cell viability. So this was exciting. We wanted to make sure that this was not a one-off just for the RAB11 locus. And in fact, we've seen that this is actually a flexible system that allows us to modify different endogenous genes. We're now knocking in GFP fused to different genes in the T cell genome, all in primary T cells. You can see that the efficiency range, but are meaningful across a range of different genomic loci. So these are the efficiencies, but we also wanted to make sure that we were actually specifically tagging different endogenous genes. And Theo did so by actually doing live cell imaging. And because we tagged different endogenous genes, the different GFP localization actually confirmed that we were getting the expected cellular localizations for each of these gene modifications. So as an aside, I think this opens up exciting opportunities now to perform live cell imaging directly in primary human immune cells as a research tool. But it also confirms that we have a flexibility to actually go in and modify or add specific sequences at different sites throughout the genome. Another critical part of this is because we're modifying endogenous genes, we wanted to demonstrate that we're not only able to modify different proteins, but that we're maintaining endogenous gene regulatory programs at these sites that we're modifying. So I first showed you the data for RAB11. RAB11 is ubiquitously expressed, and indeed we saw that the GFP was found in both CD8s and CD4s when we modified bulk T cells. But if we modify CD4 and make a GFP fusion, as we'd expect, the GFP fusion is only found in the CD4 positive cells, and in fact we see this beautiful linear relationship between GFP and CD4 staining consistent with successful specific formation of a fusion protein at an endogenous site. What I don't have time to show you is that Theo actually went on and did combinations of knock-ins. He's actually knocked in different fluorophores into combinations of 2 different genes. He's actually even pushed it up to 3 different genes that he's able to modify in a multiplex fashion. Now, I showed you some evidence that this is specific, but if we think about clinical applications, we really want to make sure that we're avoiding off-target integrations of these templates. So to really stress this, what we did is we combined the DNA template with an off-target cut in the genome, so using a guide RNA that cuts at a different site at high efficiency. And what we found was that although the efficiency of knock-in was much higher if we cut at the site of homology, we do in fact see a meaningful percentage, albeit small, of GFP-positive cells when we use an off-target cut in the genome. So we also wanted to develop a tool to actually reduce these off-target integrations, and we found that we could actually convert these long double-stranded DNA templates into single-stranded DNA templates. There's now a few different methods for doing so, and we found that if we use the single-stranded DNA templates, we could dramatically reduce the off-target integrations while largely preserving the on-target integrations. So now, with these tools in hand, we wanted to actually see if we could march this towards preclinical applications. So we started with a family. This is a family that has multiple children affected by what appears to be a monogenic form of autoimmune disease and immune dysregulation. So this is a family that's gone around the country. They were brought to our attention by collaborators Kevin Harold at Yale and others at Stanford who have seen this family that has 3 children that appear to be affected by a mutation. One child developed type 1 diabetes in the first year of life. Another child has not yet developed type 1 diabetes but has multiple autoantibodies suggesting a very high risk of type 1 diabetes. And a third actually has a different manifestation, has severe autoimmune neutropenia and ITP and is dependent on steroids. Exome sequencing has revealed the causal mutation for this family. They actually have, each parent, by bad luck, has a different coding mutation in a gene called IL-2RA, which is the high affinity receptor for IL-2 and is essential for regulatory T cells. So this family has been contemplating whether or not bone marrow transplant will be needed to treat any of these children, but they've really been looking for an alternative to allogeneic bone marrow transplantation. So we wondered whether in the future one option for this family may be actually autologous correction of the patient's own cells. And in particular, could we actually correct the regulatory T cells, restore IL-2RA expression, and potentially use corrected regulatory T cells as a therapy. So as a step towards this, we've actually been getting cells from these children sent to us in the lab. And what we found is that, as expected, as a result of these mutations, they have minimal expression of IL-2RA, also known as CD25, on the surface of their cells. That's over here. But Theo was actually able to use his method, and here we're using double-stranded DNA templates to actually correct one of these causal mutations. And we found that correcting one of these mutations was sufficient to restore IL-2RA to the surface of these patient cells. We actually also went on and tried to correct the other mutation. This is the second mutation. Here we're using a combination of single-stranded DNA template and actually the Cas9 nickase, which could potentially be used to reduce potential off-target cuts. So the combination of thinking about different template options and different Cas9 options now puts us within range of thinking about putting together an FDA package to go and see if we can actually therapeutically correct these cells and get approval to return the regulatory T cells with the gene correction to treat these children and their ongoing autoimmunity. So here what we've set out to do is correct an individual nucleotide that's causing disease. But with the ability to actually integrate large templates, we realized that we also had the opportunity to actually knock in significant new sequences into specified sites in the genome. And one of the obvious and very important places where this would have immediate therapeutic relevance is if we could actually rewrite the specificity of a polyclonal repertoire of human T cells. So human T cells naturally have a diverse repertoire of T cell receptors that allow them to recognize different antigens. But for therapeutic applications, especially for cancer therapeutic applications, we'd like to be able to define a single antigen. And this requires rewriting the TCR alpha and beta chain of a cell and specifying a combination of these two receptor pairs to give antigen specificity. So because this requires a pair that but have to be expressed in the same cell to give antigen specificity, this is actually a complicated targeting strategy. But Theo, in collaboration with Tony Rebus's lab, came up with this strategy. The idea was to come up with a guide RNA that would cut in every T cell receptor sequence independent of specificity, so cutting in the constant region of TCR alpha to eliminate the endogenous TCR alpha, and at the same time pasting in a template now that will lead to expression of both the TCR beta and TCR alpha. This is the construct that was designed. You can see that it knocks into the endogenous TCR alpha locus the full-length TCR beta, as well as the defined variable region for TCR alpha. We only have to put the variable region of alpha in, and then just the beginning of the TCR constant region, because the rest will be fused in frame with the endogenous TCR constant region, if successful. And the TCR beta and TCR alpha, are actually separated by self-cleaving peptide sequences, and the result is a polycistronic sequence if successful. So upon translation, you should get a defined TCR beta and TCR alpha expressed off of the endogenous regulatory elements in the TCR alpha locus. So this is the theory. This is the result that we had in practice. So you can see that we're actually able to define a specific sequence. Here we're knocking in the NY-ESO specific TCR, which recognizes a cancer antigen. On the vertical axis, what I'm showing you is dextramer staining for the specificity of the NY-ESO antigen. So over 12% of the cells in the experimental condition now are defined as specific for the NY-ESO antigen. The other benefit of the system is that because we're cutting in the TCR alpha constant region, we're actually knocking out the TCR in the majority of cells as a result of non-homologous end joining, we believe. So we're getting rid of most of the TCR alpha. So of the cells that remain TCR positive, the majority of those cells are now actually recognizing the defined antigen based on the TCR that we've knocked in. So we wanted to show that are these cells in fact functional, and here we've benefited from a great collaboration with Tony Rebus's lab. In vitro, we've mixed these cells with NY-ESO positive cancer cells and followed them with live cell imaging. And you can see in red and black are cells that are either have viral overexpression of the NY-ESO TCR or non-viral replacement with the TCR, and you can see that these non-viral cells kill just as well in vitro. And similarly, this is even a higher bar, they actually express pro-inflammatory cytokines and show evidence of degranulation in a manner that's specific both to the NY-ESO antigen and the MHC in which this TCR recognizes. Finally, we wanted to see, could we actually show, are these cells, do they show evidence of function not only in vitro, but in vivo? Now, the first challenge here is just, can we make enough cells that are potentially therapeutically relevant? So, although only about 10% of our cells are knock-in for the antigen that we want to create, what we showed was that easily, even in the lab, we can take a blood draw from a single individual and we can make enough cells that off of one stimulation, these NY-ESO positive TCR cells will expand, and we can easily get, in just a couple of days, over 100 million antigen-specific T cells. When we put them into a humanized mouse model with a human melanoma expressing the NY-ESO antigen, this was the experimental setup. We start with actually a mixed repertoire where some cells will have the natural TCRs, some cells will be TCR knockout, and some will be NY-ESO positive. We show that this antigen-specific cells that we've manufactured are the ones that become enriched inside the tumor bed, whereas in contrast in the spleen, we see the same ratio of cells that we put in as the input. These cells not only go to the tumor bed, but actually we find that over time, if we purify, this is a separate experiment where we've purified out the NY-ESO positive cells and injected those in, and we show that those TCR cells are actually able to control and reduce tumor volume over time in vivo. So we think that this, in the broadest possible sense, what we've developed is a platform where now we have easily sourceable reagents for rapid rewriting of endogenous sequences in human T cells. We can get guide RNAs manufactured rapidly, Cas9 protein, and now we've eliminated the reliance on virus altogether, and now we're using DNA PCR products or single-stranded DNA templates that can be rapidly synthesized and then applied for therapeutic applications. So within just a matter of 2 weeks, we can easily make a repertoire of cells that are modified in an entirely flexible manner. So where is this taking us? What we've heard before is that the genome is druggable, and now I think in the context of ex vivo manufacturing of therapeutic T cells, this is really a reality where we can go back and now look at non-coding elements and protein coding elements and have flexibility to start rewriting these programs to confer desired properties on cells for a range of different therapeutic applications. And I would just say one other thing, that not only are we within sight of actually rewriting the mutations that cause disease, introducing natural sequences like TCRs into defined sites, I think the next chapter is really thinking about synthetic biology and actually being able to rewrite endogenous sequences with newly created proteins or non-coding elements that really enhance the range of properties that we can give these cells to treat cancer, autoimmunity, infectious diseases, and really an enormous swath of human diseases. So I'll stop there. I want to thank the people who've been involved with this work. I highlighted that what I talked about today was really led by Theo Roth, but it benefited from the effort of a large network of collaborators inside the lab and a large network of collaborators from around the country that I've highlighted here and from our funding sources. Thank you.
Thanks, Alex. Floor is open. Barry?
Really nice talk. So what's the practical upper limit of the knock-ins that you can do now? Because obviously all of this sophisticated reprogramming, suicide switches, multiple cars, et cetera, will be more than 1.5 kb in the future.
We're working on it, and I think we haven't reached an upper limit yet. There does seem to be some anecdotal reduction in efficiency as we increase the size of the template. And I think that there's some toxicity that starts to creep in at a certain weight of DNA. So at a fixed weight, you're getting fewer moles of template in if the template is bigger. That said, we keep pushing the limit up. The biggest thing that we can have confirmed is this TCR alpha beta pair, where we're now knocking in about 1.5 kb, but I think we can push it even further. And I think one potential advantage of non-viral templates is that there's no fixed upper limit of carrying capacity of a virus. So we'll see.
So in the case of larger inserts, have you probed in your system the relationship between homology arm length and the insert?
We have a little bit, but we've also benefited from the work of others who have done a lot of experimental probing of this. In particular, Manu Leonetti, who was in Jonathan Weissman's lab and is now at the BioHub, had done a lot of data on this, and we sort of arrived at 300 base pairs as a compromise that's boosting efficiency but not taking up too much real estate and reducing the number of moles of template that we can get in.
And you find that to be the case for much bigger inserts too, like?
Like I said, the biggest, we're using 300 base pair homology arms for this large template that's using 1.5 kb.
Thank you.
Yeah.
When you looked at evaluation of off-target integration with a different guide, so that was an unrelated guide compared to the homology.
Yeah.
Was that designed to trap a promoter of a ubiquitously expressed gene as well?
So exactly. So there what we're using is a DNA template that has a promoter element. So it won't necessarily...
So the template as a promoter
So
in
what
that case.
we're actually showing are the percentage of off-target integrations that have the GFP and promoter integrated and expressed in the cell type. So it's a surrogate for the rate of off-targets.
Thank you.
Charlie?
The fantastic talk. I had two questions. One was, you mentioned about the difficulties of genetically expressing the Cas9 in the T cells. I was curious if you ever got down to the bottom of what the bottleneck was there. And then the second was, for the Treg therapy, what's the starting cell type you would envision using, and how long what would the durability be?
Okay, so the first question of why we can't do stable expression of Cas9, I don't know. It's something we continue to work on. I think maybe with some progress, we and others may eventually find a way to do it. I don't know whether it's an issue of toxicity or codon optimization or just not getting enough titer, but we can take the same virus and express Cas9 and infect Jurkat cells, but the same virus won't infect T cells. We get great infection with the guide RNAs, so I don't know what the answer is, but we'd like to get to the bottom of it eventually.
Just a quick follow-up on that. I mean, the mouse T cells express Cas9 fine, right?
Yeah, that's
Okay.
true. So the germline edited mice have T cells, and there we and others can get the guide RNAs in and use that. So I don't know what the issue is. The second question, for the therapeutics, with the idea would be to take regulatory T cells. You know, you could potentially also do hematopoietic stem cells, but there you have the risk of that you have to use a conditioning regimen. You may have an increased risk of causing transformation if you with off-target effects. The idea is, could we use a targeted regulatory T cell therapy? The challenge is that usually for regulatory T cells, they're isolated based on high expression of the IL-2 receptor along with low expression of CD127. So we're in the process, and we and our collaborators have found some surrogate markers to purify out the regulatory T cell population using an alternative panel of markers, correct those cells, and then purify out the IL-2RA positive regulatory T cells and give those back to patients.
How long do you expect the cells to last for?
So there's actually some evidence from phase 1 trials just using an expanded set of non-genetically modified regulatory T cells that they can last for long periods of time in the range of years, and they can be followed out. So the idea would be to have a long-lasting thing that could also potentially sort of reset the immune system and have even longer-lasting effects based on reestablishing immune homeostasis. That's the concept.
Thank you.
Great work.
Thank you.
We too have seen the toxicity of DNA, even very small amounts of DNA, to primary T cells. So the fact that you've got around that toxicity is impressive. Can you talk more about the details about the tissue culture of the cells or other conditions that you're doing to get around this toxicity?
Sure. So there's a bioRxiv preprint of the paper, and the paper hopefully will come out soon, but it's all available, and we're happy to share all the details on the experimental methods. So it's like I said, each one of these optimizations seemed to help, and by putting them together, it was sort of a series of optimizations that allowed the cells to survive this challenge. One thing that we don't fully understand that we're still exploring is maybe there's some actual protective effect of delivering the DNA with the Cas9 ribonuclear protein, and whether or not that's true and the mechanism, it's intriguing, and we'd love to get to the bottom of that.
Is the amount of DNA you're delivering maybe a lot lower than what people typically deliver?
Not necessarily.
Thanks.
Nice work, Alex. So my question relates to mispairing because we've done a very similar studies and published it recently. So knocking down track, not touching TRBC, and inserting NY-isospecific TCR. And we don't really see mispairing with that specific, very strong and optimized TCR.
Yes.
Did you measure it, and did you try with other, maybe less fit TCR? Can we rely on that?
So we haven't, we're just starting now to expand to other TCRs. The question's about mispairing with the TCR alpha and beta. We have explored some alternatives that would be used if mispairing becomes an issue. So we have two possible things that we've done. One is to combine this knock-in at the TCR alpha locus with a knockout at the TCR beta, so to reduce, and we can show that works. The other thing that we show is possible in theory is to actually do endogenous modification of alpha into alpha and beta into beta, and the possibility from multiplex actually makes this possible now.
Follow-up question on DNA toxicity. So you show you can still generate enough cells, but compared to control, what kind of fold reduction do you see?
So what we see is about 2 days after electroporation, there's a nadir of cell numbers. So and compared to controls at the same time point, there's about 60% of the number of cells at about 2 days. So it looks like about 40% of the cells roughly are dying off as a result of this treatment. But then the cells that do survive expand, and that's where we think we can make up for that cell death.
The growth curve after that point
Yeah.
looks similar.
Yeah.
Thank you.
So I have one quick question since we have a couple more minutes. Have you looked at the fidelity of the HDR products that you get with the single-stranded long templates, whether you might imagine, not necessarily true, but you might imagine that you might get some deletions or duplications, things like that.
So this is something that I think is going to be very important. There's two different protocols that we're using currently for single-stranded. One was developed by Monogenic, which is based on transcribing this doubles off with a T7 promoter, making RNA off of the long double-stranded DNA, and then doing first strand reverse transcriptase, so that you go through this transcription and then reverse transcription to make a single-stranded DNA. That you would potentially have some challenges. Are these steps all high fidelity? And I would worry about that. The other thing has been developed by CloneTech, and what they're doing is actually a PCR strategy where one PCR primer is protected from a strandase enzyme. And so there what you do is make the single-strand DNA by a degradation process. And I think that there one possibility is that you can actually sequence the double-stranded PCR product, and then the strandase hopefully doesn't actually cause any change into the single-stranded product.
Right. So I guess I was also wondering, though, let's say, assume that you have a completely pure product.
Yeah.
To what degree do you get a perfect replication of that sequence when you make the break in the cells?
So this is just an experimental challenge that I'd love to get your thoughts on. The problem is that it's really hard to do sequencing after you do these manipulations. So the problem is that the templates stick around in these cells for a long time. So it's hard to know what's being sequenced off the template versus what's being integrated. So we've actually haven't done much in the way of sequence of these integration sites, and instead have focused on protein level readouts, and are reassured that we're getting functional proteins that can actually change cell programs. But how often there's imperfect products in there, we still haven't fully assessed.
Great. Well, we've peppered you with a lot of questions. Probably need to move on. Thank you, though, for a great talk, Alex.